All right, guys, here. So yeah, so I'll give you more details on Friday, but the reading quiz next week will be mostly over the ear structures. We'll look at that, but I'll give you some more specific guidance on Friday. So yeah, so let's go ahead and dive in. Just finish these last couple things here, and then we're going to dive into the eye. So we finished last time talking about this stretch reflex here and using the muscle spindle as the receptor. And so we kind of went through those steps. The two actions were we contract the muscle that was stretched and we inhibit the muscle that's the opposite of that, or the antagonist of that one. Okay. So one thing I want to point out, we won't go through the details of this, but the deep tendon reflex is the one that deals with that other receptor we're talking about, the GTO, the Golgi tendon organ. That one is protecting us against overcontraction instead of overstretch. So what that means is the response is the exact opposite of the other one. Okay. So whereas the other one, we were worried about the muscle stretching too much, this one we're worried about the muscle contracting too hard or too much. Okay. So the response here is going to be inhibition or relaxation of the muscle that's contracting instead. Okay. And I'm going to write in, it's going to be contraction of the antagonist to that muscle. So if we're dealing with the quads again, let's say we're contracting our quads, we're pushing into a knee extension as hard as we can. Okay. If we trigger the Golgi tendon organ reflex, the deep tendon reflex, we're going to relax the quads and we're going to contract the hamstrings in with that reflex. Okay. Whereas if we did the stretch reflex, we stretched the stimulus was stretching the quads with the patellar tendon hammer, right? And then it's the opposite. And then we contract the quads on that one and we relax the hamstring. So the action is just the exact opposite. And the stimulus is different, right? The stimulus for the stretch reflex is stretch. Stimulus for the GTO reflex is contraction. Okay. And then the other one that's key that we're all very aware of because it happens to us. We've all had this experience as humans. We touch something and it hurts and our body pulls away from it, right? And we use all of our flexor muscles in that case. We step on something sharp, we flex our knee, we flex our hip, right? We flex our ankle. All those things happen at once, right? We pull away. Same thing if we touch something hot, flex our fingers, flex our elbow, flex our everything. It's all the flexors, so-called the flexor withdrawal response. So that's driven by pain, right? And we're just trying to get away from that thing that is potentially dangerous to us. Okay. So that's that. Let's do a quick question here. And let me start the clicker thing back up in the other mode. There we go. Go ahead and sorry, let me do that one more time with the option to do more than one answer. There we go. Okay. Go ahead. Try that again. Check briefly with the people on your team. Which ones do you think are true there? You can press more than one letter. So yeah, go ahead and put in the ones that you think are correct. You might need to just hit the send button just to make sure. Put your name on this. Yeah.
Right there. Like
All right, everybody just go ahead and hit the send button there. Press something. Thank you. All right, it's good. Looks like we got everybody. So we got a lot of you saying B, some saying C, some saying D. So let's walk through these. So muscle spindles sense tension. That one's wrong. Okay, that's the opposite here. So muscle spindles sense stretch. Okay, and stretch kind of feels like it might be the same thing as tension, but stretch tells us something about how long the muscle is too. as well as kind of how hard we're pulling on the ends, right? So it's not just about pulling on the ends, that's the tension one, but it tells us about the length at the same time. And so we've got stretch, and then the Golgi tendon organ senses tension, so that one's correct. Stimulation of the muscle spindle causes contraction of the stretched muscle and inhibition of the antagonist muscle. That is true. Stimulation of the Golgi tendon organ causes contraction of the one that's already contracting. No, causes inhibition of the one that's already contracting. Okay, and then we could also add to this contraction of the antagonist muscle of the one that's contracting, if we wanted to. Okay. All right. Questions on that at all? Go ahead, Riley. Yeah, for sure. So a real practical example would be throwing a ball. Okay, so when we throw a ball, we're going to use our chest muscles here, our pecs, to bring our arm forward. Okay, as soon as we let go, or as soon as we let go of the ball, we need to start slowing down our arm. Okay, so really, what happens if our reflexes are trained for this is we're contracting, contracting here. Right when we let go, our GTO fires, our pecs start relaxing. So that's the agonist. That's the one that was contracting, right? And now we contract our antagonist of those, which is going to help slow our arm down, right? So that's kind of a real practical example of how it works and that kind of activity. So it's a relaxing the one that's contracting and then contracting the opposite of that muscle. A couple things would happen. One thing that would happen is we would rip our muscles right off of our bones because our muscles are always more powerful than we can ever use them. And so, for example, like when someone has that tetanus infection that we talked about last semester, that overrides these reflexes, and we can break our own bones with our muscles because they're so strong. So this is not only helping us with movements like I just talked about, but it's also just kind of limiting the amount of force that we produce with our muscles. Unless we're in a really stressful situation, then we can override that as well and do things like lift a car off a kid and stuff like that. But that's going to hurt us later. But we always have more power in our muscle than we ever use. Yeah. So yeah. Question. Okay, so last thing on reflexes here before we jump over to the eye is we can have pathologies or just kind of differences in reflexes, and those can be that we're extra reflexive, right? We have big reflexes, that's hyperreflexivity, overactive reflexes. This is typically caused by your brain not getting involved in the reflex, okay? And so a lot of times that's because your brain isn't working so properly, okay? So that could be alcohol, that could be drug use, that could be just distraction, or it could be a spinal cord injury where the brain can't actually connect to that part of where the reflex is. So then we have much greater reflexes because our brain isn't able to modulate them at all. And then the other side of things, hyporeflexivity. This usually is a problem when we have damage to our nerves. So it's just like a slow response going to the spinal cord and back. And that could be the type 2 diabetes where we have some neuropathy, right? Some damage to our nerves as well as other nerve damage problems. So. Okay. All right. So we're going to jump over to the eye. Let me pull that one up here. And I kind of moved some of those slides about questions to another day because I knew we'd be running short on time. So we'll get to those at some point. But if you're wondering why we're not doing those in your notes, we'll get there. Okay, so let's jump into the eye here. So this one is mostly about the structures of the eye, and then Friday we'll dive into the kind of physiology and how they all work. So just some fun facts about the eye. The eye contains 70% of the body's sensory receptors, right? So we have something like I have it in my notes here, 385,000 sensory receptors in our whole body. And 70% of those, 105 million of those, are in these two little spots in our head, in our eye, right? Almost half of the big part of our brain, our cerebral cortex, is involved in vision in some way, right? So we're very vision dominant organisms compared to dogs and cats and other animals are very, very chemical sense dependent, right? So much more reliance on their nose, whereas we are much more reliant on our eyes. Okay, so just some basics on the structures here, right? So we got our eyebrows, okay? Eyebrows are helpful for shading our eyes as well as catching sweat dripping down. We've got our eyelids, and our eyelids blink reflexively, something like every 3 to 7 seconds. And the whole purpose of that is to spread lubricant from kind of the top of our eye down to the bottom and across as well. So, and that also can happen reflexively when something's coming towards our eye. Our eyelids will close reflexively to protect from something hitting our eyeball. And then we've got our tarsal glands here, and we can see those just right up above the eye here. And those help to secrete some of those lubricating fluids that we're trying to get across the eye to keep the eye moist. And so you only really need to know the ones that I've highlighted there on that image. Okay, so one other important structure in our eyes are called the conjunctiva. The conjunctiva are the mucous membrane that lines the inside of our eyelids and the white portion of our eyeball. Okay, and so this is very important for releasing mucus as well and keeping our eyeballs moist. Okay, so when this structure gets irritated, this is called conjunctivitis, right? And this is pink eye, right? And so when somebody has pink eye, if that conjunctivitis is caused by a bacteria or virus, that's when we typically call it pink eye. And it can be highly contagious for sure. Some of you know. Karen? Pink eye is kind of the name for conjunctivitis caused by bacteria or virus. Yeah. You can flare up your conjunctiva with dust or with other things that get in there, but if it's caused by a microorganism, then it's typically called pink eye. Yeah. Either way, your eyes are going to be red and pink, but that's just how it's used. Yeah. Questions on that? Okay. So then this whole setup with this system called the lacrimal apparatus, this is how we produce tears. And so the lacrimal gland is located up here in kind of the superior lateral part of our orbit, and it releases tears. And there's little ducts that connect right in here and drop the tears in the upper right or upper outside part. And then with blinking, that can help it move across and down over to our nose. And so yeah, so tears contain a few different things. So they contain mucus, which is helpful for, again, moistening, lubricating, protecting our eye. Also some antibodies. This can help destroy microorganisms that get into our eye. Yeah, Ava? Yeah, it can be related to the content of the tears. And if your eyes are already a little bit irritated for some reason, and then with that extra fluid put on them, that can cause some burning. Um, We also have the makeup of tears can change depending on whether it's emotional crying or just tears, and we'll talk about that at the bottom here. But some of those things could cause a little bit more stinging than others, depending on the state of your eyeball. But we also have lysozymes in our tears as well, which is also helpful for specifically destroying bacteria that might have gotten into our eyes. So all of this is really helpful for just maintaining the health of our eyeballs. And these secretions are all stimulated to be released by the facial nerve. Yeah, Caitlin? Yeah, so bacteria, fungus, viruses, those kind of things. Yeah. Mhm. Yeah. And so really the kind of the pathway of the tears is being released from the gland up the top there, going through those little ducts across the eyeball, into the lacrimal sac, which is right here. And then that actually connects into your nose, right? So this is why you get a runny nose when you cry, right? Is because the excessive tears go through that little duct and they dump out into your nose, right? And so in newborn babies, it's super common for this to get clogged up. And you see babies with like crusty little eyes. It's because that's kind of clogged up, and they tell you to like stick your finger in there and kind of smash it around and try to loosen it up. So they'll continue to flow. But not too far, stick your finger right in their eyeball, but yeah. And so then we normally have kind of a steady flow of tears, but then with emotional crying, we get a lot more. And the interesting part about that is different components are added to the tears when we have emotional crying. That's my least favorite part about this classroom is you guys in the back row. So sorry about that. Any that time I have stuff on the bottom of the slides, they have to stand up in the back to see it. So sorry about that. Yeah, so enkephalins are a neurotransmitter that we talked about last semester that are the kind of the intrinsic version or the version of our body that makes it similar to opioids, but the intrinsic version of those. And so those are released in our tears when we cry, and so it's like a feel-good neurotransmitter that helps us feel better. Yeah, and we talked about this blockage here as well. This can lead to mucus buildup and tears in your eyes. Okay. Questions on any of that? Okay. You go ahead, Ethan. Yeah. Absolutely. Yeah. It's soothing for sure. Yeah. Yeah. It's definitely soothing. It's like relaxes your stress from whatever stressful situation you're dealing with. Yeah. Yeah. So, so don't hold it back. Let it just go ahead. You can cry. It's okay. Okay. All right. Let's take a sec and just kind of do this little recap here. Go ahead and fill this in with your team. So where do we find these things? What are the functions? We'll just spend a couple minutes here and then we'll move on.
Okay, so eyelids, they like, I guess they protect bacteria. Lubricate. Do they just, does that have use for? Yeah, it's like, it's on your eye.
The mice sometimes kind of like the fences on our skin. Do they like do the same?
Very similar. Yeah. Mhm. Yeah. That's hot back here. Huh?
It's gonna click on them.
Mason's wearing the right shirt. That's really hot. It's like cool up there.
The nice thing is though, if you break from this, you get a view of.
Very nice. Yeah. I can't complain. Yeah. All right, guys, let's come back together. I don't necessarily need to write all this in. I think it's all there, but any specific questions about this before we move on? Okay, so we'll kind of have, if you didn't have me last semester, we'll have little things like this, just kind of break up lecture, a little brain break where you kind of recap something, kind of help you solidify it, then we'll move back on from there. So always a good idea to go ahead and write stuff in, even if you can tell it's going to uncover. If you need to change it a little bit when you see the right answer, that's okay. But the educational research is real clear that making a decision on something, whether it's right or wrong, helps you learn it more than just waiting for me to uncover it and then you write in exactly what you see. That's just kind of, you don't really learn it that way. So that's why I do a lot of this stuff with the clickers and this kind of stuff. So, okay, so let's talk about some more structures in the eye. Okay, so you've familiarized yourself with this through the reading and through the getting ready for the reading quiz. So we have 3 different kind of layers in the eye, and they're called tunics. Okay, and so the very outer layer is called the fibrous tunic. The white part of this, the posterior portion, is the sclera, and the sclera is tough and thick and strong, and this serves as an attachment site for muscles, and it gives the eyeball its shape. And then the cornea is the very anterior part of this, has a little bit of epithelium on the internal and external surfaces, and then it contains a very special protein that's totally transparent. Those are called crystalline proteins, and that's what allows the light to go through. And the function of this, of the cornea, is definitely to protect, so things can't get through and get into the eyeball. But then secondly, to bend the light as it goes through. So this is kind of the first bending of the light as light passes through the eye. We get a good portion of that bending happening by the cornea. Then we're going to get the kind of fine tuning of that later by the lens, but the cornea is important for the initial bending. Okay. Got it all back there. Last semester, I actually asked them to try to move everything up a little bit so you guys could see a little bit back there, but they never did anything, I don't think. But because I could try again, see if I can just point the camera a little bit higher, that might help. But okay. All right, so the middle layer, middle tunic is called the vascular tunic. And that one is the choroid, like you guys already looked at with the quiz, okay? This is the posterior region of this, has lots and lots of blood vessels, and then cells that contain pigments, and those are proteins that absorb light, right? Pigmented proteins are proteins that absorb light. And the function of all of this is to, number one, absorb light, which helps us to minimize light scattering. And then it supplies blood too. All the blood vessels are supplying blood. Okay, so when you've heard the term a deer in headlights, right? If you grow up somewhere where deers cross the road in the middle of the night and they literally just kind of freeze if they look at a car coming at them, part of that is because they don't have a very thick choroid layer in their eyeballs. And so when bright light goes in their eyes, it just scatters so much that it knocks out all of their rods and cones. So they can't see anything, just kind of blinds them for a second. So human eyes have a little bit more protection against that because of this thick choroid layer to help absorb a lot of that extra light. The ciliary body is the anterior region of this middle vascular tunic. It's smooth muscle, and it is under control of oculomotor nerve 3, and this is what changes the shape of the lens. And we'll talk more about those details and how all that works in a sec. And then lastly, the ciliary processes, which I'll read that to you for the ones in the back there. This is just what secretes that aqueous humor that we were just talking about. Ciliary processes secrete aqueous humor, and that's the stuff that's in the front of the eye, the anterior chamber of the eye. Okay, so aqueous humor secretion for the anterior chamber of the eye. So the lens is kind of the dividing line between anterior and posterior chambers, right? And so everything in front of the lens is anterior, everything behind the lens is posterior. Okay. All right. So then we'll continue on here with this middle vascular tunic. The other part we haven't talked about yet is the iris. Okay. So the iris is made up of two different muscles, and the muscles themselves are pigmented, and that's what determines the color of our eyes, right? And so the function of our iris is to change the size of the pupil, right? And dictate how much light comes in. So by contracting and relaxing these two muscles, we change the size of the hole, which is the pupil, right? And change the amount of light that comes in. Okay, so the pupil is just that, it's just a hole. And so that lets light into the eye, but the iris muscles are the ones that decide, kind of dictate how much gets in. Okay, so we've got two muscles in the iris. The one on the outside, this one here, you can see the fibers are directed this way. Okay, that's called the pupillary dilator muscle. And that one is controlled by the sympathetic nervous system. Okay, so this one is SNS. And then we have the pupillary sphincter muscle, which is controlled by the parasympathetic nervous system, PNS. And that's a sphincter muscle, so that means it's round. So when we contract it, it shrinks down the hole. When we relax it, it opens up the hole. Okay, so with normal changes in the size of our pupil, like happen when we go from being in a going bright outside to then a darker room, our pupils are going to get bigger, right? Because we're trying to get more light. If we go the opposite, if we go from a dark room to the outside, our pupils need to get smaller to block some of that light, right? The normal control of that is all about this pupillary sphincter muscle. Okay, so it's when we want to make our eyeballs smaller, so pupil constriction, our pupils smaller, we use our oculomotor nerve to activate our pupillary sphincter muscle, and that causes constriction. And this is letting less light in. This is also what we do when an object gets closer to us. We shrink down our pupil. We'll go through some steps related to that next time, but that's just a preview here so far. And then for pupillary dilation, what all we need to do is we need to inactivate that muscle. Okay, so instead of contracting it, now we're relaxing it. So we're decreasing the amount of activity on oculomotor nerve 3. We're relaxing that pupillary sphincter muscle and we're dilating it. And this would be, that just says far vision, more light. So just the opposite of this one, far vision, more light on the bottom there. Okay. So we haven't really talked about the sympathetic one yet. Okay, because this is really for the vast majority of the time, this is what we're doing to constrict or dilate. Okay, it's only under very stressful situations that we activate this pupillary dilator muscle, and that one pretty much just pulls open the other one, right? It just rips it open. And that's kind of under stress or pain or things like that, when we don't really care about how clearly we can see something. We just want to see as much as possible, right? So we open up our pupil so we can kind of take everything in. Doesn't matter if it's perfectly clear, right? We just want to see that dog running at us or whatever it is that might be coming from the side, right? So questions on that? That makes sense? Okay. Yeah, let's just kind of walk through this one together here. And so, yeah, so we can kind of here under situations of stress, we dilate our pupils via, okay, we can say contraction and relaxation of the pupillary dilator or sphincter muscle. So if we're talking about stress, then which nervous system are we talking about? Sympathetic or parasympathetic? Say it out loud. Sympathetic, right? Fight or flight response, okay? So then we need to use that dilator muscle here, okay? And we need to contract it. Okay, so when we look here, again, this dilator muscle is pulling open like that. It's just ripping open. So all those little fibers are shortening and we're pulling that open. Okay, but under situations where we need to see something far away, we still need to dilate our pupils, but it's not a stressful situation. I'm just going from reading to looking out there. That's not stressful. It's not painful, right? So instead, we just relax our pupillary sphincter muscle, and that's what opens up our pupil. So kind of two different ways um, to dilate our pupil, right? One under stress, one not under stress. That makes sense? Questions on that? Okay. All right. So let's take a sec in your teams again and just label these structures we just talked about in these different parts of the eyeball. Hopefully you guys are already familiar with this. Fill it in if you haven't already done that. And let's send in this letter on your clickers there. Okay. Yeah, so go ahead and send in that question on your clicker there.
How much focusing happens to the light?
Mhm.
Would that mean like, what would you
Changing the lens.
changing?
It's okay. I just need to remember to get some more so I have some backup. But yeah, no, it's okay. That's fine. Yeah. I just need to remember. So I got some backups for somebody else. But yeah, you're good. You can keep those. Yeah. All right. Send in that answer on your clickers there. Everybody go ahead and send in something. So the structure in our eye that we haven't really talked about yet that controls how much the light is focused is the lens, which is not labeled on this one. That's this one. But the muscles that control that are here, right? And so that was E. Okay, so you can see all these structures here. So yeah. So the ciliary body has kind of two parts to it. It has the smooth muscle that changes the size of the lens, and then it has the part that releases aqueous humor as well, like we talked about. So yeah, we haven't got to that part yet, but that was just kind of a preview thing there. These are called the suspensory ligaments that connect the muscle to the lens. We'll talk about those functions in just a sec. So E here. Okay, questions on that? Go ahead. Yeah.
Could you explain why the muscle that controls how much focusing happens is the ciliary body and not the iris? 'Cause I thought it was.
Yeah, so the iris dictates how much light comes in, right? And then the one that changes the shape of the lens dictates how much we focus that light after it gets in. And so the iris is controlling just amount of light. Lens is controlling how much it gets focused.
Oh, okay, got
Yeah.
that.
Good question. Yeah. Good. Other questions? Okay. All right. Well, let's keep going here. So then finally to our last tunic, our last layer called the inner or sensory tunic. This is really mostly the retina. Okay. And there's kind of two layers to this. The pigmented layer is the layer that's right up against the choroid. And this is, again, more pigment to help prevent light scatter, as well as pigmented proteins there available to be put into our photoreceptors, our rods and cones, to replace those ones that were damaged. Okay. And then we have the nervous layer of the sensory tunic, which is where we have our rods and cones. Okay, so those are our photoreceptor cells. Rods are activated by low light and only give us information about black and white light. But then cones are stimulated by stronger light, and we have three different types for different colors of light. And so this is where we get non-black and white images. Okay, and then we got the bipolar cells, which are the ones right in the middle, connecting the photoreceptor cells to the ganglion cells, and then the ganglion cells are last. And it's the axons of those ganglion cells that make up our optic nerve, right? So we get an action potential on the ganglion cells that goes through the optic nerve and up to our brain. Okay. So we can kind of just walk through this labeling one together, since we just talked about it. Okay, so A is going to be an example of a rod. Okay, whereas B is an example of a cone. Okay, and we can see this is the pigmented layer right here, right? That's that other layer. Sorry, that's really bad. But pigmented layer there that everything is kind of attached to. So that provides replacement pigments for the photoreceptors. And then we move on to C. Those are the bipolar cells. That's kind of this connecting point between the two types of cells. And then D, finally, the ganglion cells. And E is just a collection of all the axons of those ganglion cells, but that's the optic nerve. The kind of interesting part about this is that light comes in this way, right? But then the signal goes this way, all right? So it's like light passes over all the cells. I'm hitting arrows here. Yeah, Karen, go ahead. Oh, what I wrote, yeah, I said pigmented. Yeah, so that's the pigmented layer of that inner vascular tunic. It's just that little like kind of strip of tissue before you get to the photoreceptor cells. Other questions? Okay. All right, so that's kind of the details of the retina. When we look at the retina at a larger scale, we have three primary regions to it. Okay, so we have what's called the fovea centralis. The fovea centralis is the area where we have all of our cones. Okay, we can see the three regions here. Fovea centralis is directly behind our pupil. Okay, you'll notice that the optic nerve is not directly in the back of the eye. It's a little bit offset. Okay, because that light comes in and most of it's going to get focused to land right there on the fovea centralis. And the reason and the way our pupils set or the way our retina set up is that that's where we have almost all of our cones. So that's the whatever lands there. We're going to see that image very clearly. We're going to see higher, stronger light images there because that's where all of our cones, all of our colored images show up from the fovea centralis. And then we have the blind spot, which is right here because this is all just loaded with axons. There's no photoreceptors right here, right? So you guys will, I think, still do an activity in the lab where you kind of find your blind spot. You kind of keep doing this for a little while, and then eventually your finger will disappear, and that's because the light is hitting that spot right there. So I think you'll still play with that in lab. And then lastly, the periphery. That's just the rest of it. So that's all of this, right? And the periphery is where we have mostly rods. There's still a few cones there, but mostly rods. That's where all of our peripheral vision comes, right? When we're looking at something straight ahead, we still kind of have awareness of stuff over here and over here. It's not very clear, right? But we can still kind of see it. That's all the light hitting our cones, or sorry, our rods in the periphery of our retina. Okay, questions? Okay, so then talk a little bit more detail about the lens. And so the lens, again, it's the dividing line between our anterior and posterior segments. It's very elastic and we can stretch it out. And then when we let go of it, it returns to its original shape. Okay, and really our ability to see up close is all about the lens's elasticity and its ability to go back to a very convex shape. Okay, so this is what does the final, sorry, this is what does the final focusing of the light that we see. Okay, and so it kind of changes the degree of light bending through refraction to try to get the focal point to land right on that fovea centralis in our retina. Okay, so then we look at what's behind in the posterior segment behind the lens. This is called vitreous humor, and this is kind of like jello-like substance that is pretty thick and solid. It's transparent, so the light passes right through it, but it maintains the shape of the eyeball. It keeps the retina pushed up against the eyeball so it stays on there. And it's really only formed once. So if your eyeball gets ruptured, if you have a globe rupture, then it takes lots of surgeries and other things to kind of, I think they have some kind of a synthetic version of this thing kind of put in your eye to maintain that pressure to hold your retina in place. Because without this vitreous humor, your retina kind of falls off of the back of the eye. It's called retinal detachment. When I was working as an athletic trainer for the baseball team here, just a fluke accident. We had one of the base there just during practice. The guy just ripped a line drive. They had the thing up to try to protect the people like outside of the base path, but the coach was just like right just in the spot where he could, like the first spot he could get hit by it. The line drive hit him directly in the eyeball. And it pretty much did that to his eye. That's why I have that picture. This is not his eye, but I found one, but it just ruptured his globe like that. And so everything just gets kind of shot out the back, including the orbit. So orbital fracture all around, and then shot all the vitreous humor right out the back. And so he had something like 7 surgeries to try to fix his eye. And he was able to see some, but definitely never had his full vision back again after that. So yeah, picture just reminds me to tell that story. But okay, so that's vitreous humor. On the other side of the lens is the aqueous humor. Okay, so this is the more liquid-like stuff. So plasma-like, as we saw in the quiz. This we're constantly replacing. So we're draining it and putting new stuff in. And this is all happening by those ciliary processes. And the function of this is to give nourishment to the lens and the cornea. We don't have a real direct blood supply to this part of the eye. So we need to get oxygen and nutrients into that aqueous humor so that can help supply the nutrients needed to the cornea and the lens. And so when someone develops glaucoma, glaucoma is typically a problem with the drainage of aqueous humor. And so we're trying to reabsorb the aqueous humor to kind of replenish it because we're making new stuff and putting it in. And so if we're not able to reabsorb it quick enough and we keep adding more, then we're building up pressure in this, and that shifts the lens back, pushes everything backwards, and can damage the optic nerve with that pressure. And so this is super common in type 2 diabetes because the little pathways for the aqueous humor to pass through get kind of clogged up and with plaque and stuff. So yeah. Questions on that? Okay. All right. So then This is kind of a review from last semester, so I won't spend a ton of time with these extrinsic eye muscles, but let's run through them here. And so we see we have the four rectus muscles, right? So we got all that info there. So the superior rectus is on the top, moves the eyeball up, and this tells you which cranial nerve controls it. Okay, inferior rectus moves the eyeball down, also cranial nerve 3. Medial rectus moves the eyeballs in or medially towards the nose, also cranial nerve 3. Lateral rectus moves them out. Cranial nerve 6, called abducens, right? Because it abducts the eyes. Okay, and then we got the two obliques. Okay, superior oblique and inferior oblique. Superior oblique does this, so it pulls the front of the eye down and out, whereas the inferior oblique pulls the front of the eye up and out. Okay, so we've got cranial nerve 4 for this one, which is called the trochlear nerve, which is named after the little pulley-like ligament that this muscle runs through. And then again, cranial nerve 3 for the inferior oblique. Okay, so you guys remember all that perfectly from last semester, I have no doubt. Right? We're all good? Give you just a sec to finish writing that. Okay, and I'm gonna let you guys fill this in on your own, not right this second, because I want to spend the last few minutes talking about the tracking part. But if you have any questions on it, you can always come back and ask me, but it's just labeling those muscles here. Okay. Yeah, real briefly, this is that trochlea thing, that trochlea where the superior oblique goes through. It's a weird little pulley system set up there in the eye. Okay, so we can kind of just briefly look at this, which is when we track an object upwards, we don't need to worry about filling in the obliques here, but tracking an object upwards, which muscle are we going to use? Superior rectus. Okay, downwards, we're going to say inferior rectus. Okay, track an object to the right. Now we have to say that do the right eye and left eye separately, right? So to the right would be right eye, would be the lateral rectus, and then left eye would be the medial rectus, right? Because our right eye is going out and our left eye is going in, right? As we go this way, okay? And then just the opposite here. So our right eye would be medial rectus, and our left eye would be lateral rectus. And then if it's moving towards you in both eyes, we're going to be using the medial rectus. If it's moving away from us in both eyes, we're using the lateral rectus. Okay. Okay, questions on that at all? Sorry, these first couple classes, there's always a lot of content and we're always doing all the extra stuff too, so we have to kind of rush, but we'll get caught up. I want to finish this one today so we can be on track for Friday. So, all right, last thing here, how our eyes move. Okay, so this type of movement here called saccadic movement really means that our eyeballs kind of jump from focal point to focal point. So for example, go ahead, you can do this as well, like look to your right, focus on something on the wall, and then just slowly start moving your head all the way around. You'll notice that your eyes stay on that focal point, and then until they can't anymore, then they jump to another one. Then they stay on that one, and then they jump to another one, right? And they jump to another one. That's just what our eyes do. They want it, if they just scan across, they can't focus on anything, right? So they have to stay on indistinct or distinct focal points. And so that's what saccadic movement is. It's different if we're tracking a moving object though, right? So if I take my pen and move it, then my eyes can stay on that, right? Because it's one focal point really, right? That's different than multiple things that we're looking at, right? So that's the tracking one. And then as far as some abnormal movements go, strabismus is just a medical term for what we would say cross-eyed, right? And this oftentimes results in diplopia, which just means double vision, which is just pretty much your two eyes are looking at two separate things. So we're getting two different images in our brain, right? Which is problematic. Your brain creates two images, right? And you're trying to figure out which one to look at. So yeah. All right, guys, we're good. We finished. Have a great rest of your day. We'll see you guys on Friday. Have a good lab if you haven't been there yet.
Thank you.
Bye, Brooke. I don't see you, man. How's the elbow doing?
Thank you, Dr. Strong.
How's the elbow? Yeah, yeah. I'm starting to get any light. Okay.